You have a thin filament, what's that called? Actin, and the thick filament is myosin. Do you guys notice the colors here? Which one is darker? Thick or thin? Thick. Which one is lighter? The thin one. What do muscle cells look like under a microscope? What's that special term we used to describe them? It starts with an S. What? Striated. Why do you think they're striated? It's because of this arrangement here. Thick, thin, thick, thin, thick, thin. But here, good? All right. Okay, let me get through until this slide here, and then I'll give you guys a break, and then I need somebody who is okay getting zapped today. Okay. All right. The contractile unit of a muscle is called a sarcomere, and sarcomeres have specific borders. They will always run from Z disc or Z line to Z disc, so Z disc to Z disc. I specifically put two different pictures on here for you because one shows a particular protein and the other one doesn't, and vice versa. Okay, so your myofilaments, when you're looking at them in a sarcomere, are always going to be very, very ordered, but they will overlap slightly. Now, can I ask you on your lab practical or on your exam, what is the contractile unit of a muscle? What is the answer? Should you know the parts of a sarcomere? Okay, so your Z disc or your Z line, that is the border of your sarcomere, and that's going to be located entirely within a band called the I band. The I band, when you're looking at it under a microscope, is what appears light in that micrograph of a skeletal muscle because the I band contains what filament only? The thin one, which is, what's the name for it? Actin, okay? So the I band contains actin only. And in the middle of that I band is the what line? The Z line or the Z disc. Everybody here clear? How I remember this is Z because it kind of looks like a zigzag. Everybody here good? Okay. The A band is where you have myosin with some actin overlap. Because the A band contains myosin, that is what appears dark in a micrograph. That's what gives you your striations. Everybody here clear? But do you notice that within the A band, right smack in the middle, there is a portion where you have myosin only. Does everybody see that? That is called the H band. So the H band is always in the middle of your A band, and that's where you have myosin only. Everybody here clear? Right smack in the middle of that H band is something called the M line. M stands for middle, okay? And why am I pointing that out? Because, well, M could also stand for myosin, right? But it's the middle. It's because when we go through sliding filament theory, these actin molecules right here, the ones in red and the ones in purple, will move towards the center. And what's in the center? The M line or the middle. And when it moves towards the center, what happens to your zone of overlap? Does it increase or decrease? They increase, so which band is going to disappear? The H band is going to disappear. Does your A band change in length?
No.
Serena, what did you say? No. Your A band should not change in length. How do I know that? Does your myosin change in length? No, it doesn't. Your myosin length stays the same. So however long the myosin is, that's how large your A band is going to be. But what's going to disappear within the A band? The H band. Because what happens? Your zone of overlap increases or decreases? Increases, and where is it moving towards? The middle or the center. And what's in the middle? Your M line. Everybody here clear? Yes? Okay. So a couple of things here. Your Z line or your Z disc is going to be made up of a special protein called actinin. And then looking at this picture right here, okay, looking at this picture, do you see how here's the myosin? And at the ends of the myosin, there's kind of a spring-looking thing right there in green. So that is a protein called titin. So titin anchors myosin over to the Z discs. Think of it as a tether, okay? So So if you run, and you have a string attached to you, how far can you go? Not very far, and then eventually what's it gonna do? It's gonna pull you back, right? So that's the same thing that's happening here. When these guys, when the myosin heads pull the actin towards center, it's gonna pull on those strings and it's gonna produce tension. That's what your contractions give you. So if you, again, do your calf raises, I am contracting my muscles right now. How am I producing tension? Tethering on that tighten. Okay, notice the spelling of tighten. What does that word sound like? Tighten. Tighten with what ending? Tighten, like the regular word. A-N, Titan, like Titanic. What does Titan mean? What does Titan mean? Okay, not Titan like your shoelaces, but Titan like Titanic. It's massive, it's huge, right? Okay, that's not a coincidence that they named this Titan. This is the largest protein in the entire human body. You should write that down because I can ask you that on your test. What's the largest protein in the human body? Titan. Okay. If you spell this Titan on your test, what am I going to tell you? You're wrong, because Titan is, you know, if you know anything about Greek mythology, they're the parents of the Olympians. Okay. They're the parents of the Olympians. And then it also means really big. But is that the protein in your muscles? No. Titan, I-N. I'm going to be a pretty big stickler when it comes to spelling on this lab practical in particular, okay? Specifically for titin, nebulin, actinin, and sphygmomanometer. Yeah?
It's the largest one?
Largest protein in the human body. Everybody good on that? Yes?
Titan, nebulin, and
Titin, nebulin, actinin, sphygmomanometer. Okay? Okay. Okay, we need to talk about actin and then myosin, and I think after myosin I'll give you guys a break, and then I need to zap somebody here. Who would like to volunteer as tribute? Nobody? Anybody? No?
I can do it.
Okay. It might hurt a little bit, just
It's
telling you.
fine, yeah.
Okay, alright. You sure?
Yeah.
Alright, so your actin filaments, which are your thin ones here, if you look at them, these guys are actually comprised of two strands that are intertwined with one another. So Mardi Gras coming up, right?
Mm-hmm.
Kind of, I think it's like February 21 or 23 this year, something like that. Okay. So I want you guys to think of Mardi Gras beads. Notice how those kind of look like beads on a string? Okay. So that's actually how actin looks like. So I want you to notice, here's actin right now. Each individual bead is what we would call G-actin. G stands for globular. Notice how they look like little beads. Everybody here clear? Okay. When you have a bunch of G-actin together and kind of looks like a string, this string is now called F-actin, F for filamentous. Because what's a filament mean? Like, what's a filament? Oh, you guys, come on, you're killing me here. What's a filament? You guys have heard of filaments before, right?
Mm-hmm.
What's a fiber? Guys, this is a little painful for me. Come on, what's a filament? What shape do you associate with the word filament or fiber? Like a what? Like a line or a string, okay? So each one of the beads, this is globular actin, that's why it's called G-actin. If you have a bunch of globular beads together, then it forms a string. What is that called? F-actin. What's F stand for? Filamentous actin, okay? So your actin filaments is actually two F-actins twisted upon each other. Everybody here good? All right. So looking at this picture here, do you notice that there's kind of a thin blue portion right smack in the middle here? That is a protein called nebulin, and nebulin keeps your F-actins together so they don't come apart. So think of nebulin as kind of like glue. Everybody here good? Okay. when you look at, and this is an oversimplification, okay? I am not an artist, so I can't replicate this, but I want you guys to see here, there was an attempt. Each G-actin has a myosin binding site associated with it, which is what I'm gonna say is an orange up here. You guys see that? Notice how here in this picture, they're kind of like in a little yellow. Everybody here clear? But notice how it's blocked. Does everybody see it? So in the same token here, these myosin binding sites on these G-actin are also blocked. What's blocking it? Tropomyosin, which is your gate. Hey guys, if that myosin binding site on actin is blocked, what can't bind to it? Here's a hint, it's in the name. Myosin. Myosin can't bind to it if it's blocked. Okay, well there's a gate. What do you think we're gonna have? Oh, we have other proteins associated with it. And I hope you guys don't mind me showing it to you here because it's really kind of difficult to see here. Notice how you have the tropomyosin, which is easy enough to see. And then there's another protein, set of proteins here that's kind of like in this brown color that almost looks orange too. So I just redrew it up here, okay? So associated with tropomyosin and actin is another set of proteins called troponin. Troponin, you're gonna find out in your lecture set today, has 3 subunits. Troponin I, troponin T, and troponin C. Troponin I means inhibitory, meaning that's what's gonna keep the tropomyosin in place. Troponin T stands for tropomyosin, meaning that's the part of troponin that binds to tropomyosin. And then troponin C, what do you think troponin C stands for? What's the C? Calcium, because what does calcium do? Oh, it's your key. So as soon as troponin C binds to calcium, troponin is gonna undergo a conformation change that will then allow it to slide tropomyosin out of the way and expose what? The myosin binding sites. And once you have the myosin binding sites exposed, then you can form something called a cross bridge where the myosin heads will bind to the actin. Did I explain this clearly enough to everybody here? Okay, from this slide, you need to know the structure of actin, know the difference between G and F actin, know what nebulin is, know what the roles of tropomyosin, troponin, and calcium are. What's troponin? What is its role? What's the analogy? It's the gatekeeper, so that means it's gonna move what out of the way? The tropomyosin, which is the gate. But how does it move it out of the way? What does it need to bind first? Calcium. Good. Okay. So here's myosin. The reason why myosin is so thick is because it's got 6 chains of proteins associated with it. You have 2 heavy chains and 4 light chains. And then they'll basically twist together to form something that looks like golf clubs. But the part that I really need you guys to know and understand here, I think after this slide or the next slide, I'll give you guys a break, okay? I need you guys to mark, star, highlight this portion here where it says myosin heads, okay? So the myosin heads are incredibly important because they're gonna have 2 main binding sites, okay? One site will bind actin, hence the reason why you have this, okay? So the myosin head has 2 binding sites. One binds what again? Actin. The other one is gonna bind ATP. So write that down. It's not on here, so I'm telling you right now to write it down. Your myosin heads will bind 2 things. What's the first one? Actin. What's the second one? ATP, okay? When it binds ATP, that then is gonna have another feature associated with it, which is an enzymatic activity. It's called myosin ATPase. Myosin ATPase hydrolyzes ATP into ADP and in inorganic phosphate. That's not pi, that's inorganic phosphate. I really should have made that subscript there. An inorganic phosphate, okay? So here's what I want you guys to understand. A lot of people think that ATP is required for muscle contractions. It is not. You need ATP for relaxation. What you need for muscle contraction is ADP, diphosphate. You cannot get ADP unless you can hydrolyze it. That's why this myosin ATPase activity is incredibly important. Does this make sense to everybody here so far? Okay, so I'm gonna give you guys an analogy and I'm gonna ask a couple of people to help demo this for me later. I want you guys to look at my marker here, okay? I want you to think that this entire marker is ATP, adenosine triphosphate. But what can I do with this marker with my hand? Can I uncap it?
Yeah.
Okay. Notice how I'm still holding cap and marker in the same hand here. This is an ADP, the marker, plus inorganic phosphate marker cap configuration if I'm holding it in the same hand. Does everybody here see it? This is a hydrolyzed form. Notice how the marker is not cap. Everybody here clear? If I get rid of the cap, what's left in my hand? ADP. But whenever I get rid of the cap, the phosphate, that releases energy, which your muscle cells, the myosin is gonna use to undergo something called a power stroke, which is where it's gonna pull actin towards what? And towards the center, towards the middle. And what band is gonna disappear again? H band. Does that make sense to everybody here? Okay, so here's a question for you. What do you need to bind the myosin to the actin?
ADP.
You need ADP. You need ADP. Whether it's ADP by itself or the phosphate group, doesn't matter. You need ADP present. What do you need to relax it? ATP. So I have a question for you. I have a mutation where the myosin ATPase activity of my myosin heads has been rendered inactive. How would that affect muscular contraction? Talk to each other.
Can you repeat the question?
Yeah. I have a mutation where the myosin ATPase activity of the myosin head has been rendered inactive. How would that affect muscle contraction?
So you want me to edit that?
Talk to each other. I should hear talking. I'm gonna go around and I'm gonna collect your answer sheets,
I wonder,
okay?
but it wouldn't be would it be,
We were more than 10 minutes late, so I gave you 1 point. Your name is on here? I have a stapler up front. Well, why not? Oh my God.
Like, would you feel a difference?
So if that's not.
You know, like, you get a cramp, like, oh my God.
Okay, I have a question for all of you. What's gonna happen to muscle contraction? Go or no go? No go. All right. So here's another question for you. If you tell me it's a no go, do you think that will be enough to get you credit on your test? No, because you didn't explain it, right? So what's the explanation? Why, Liz? Because I heard you. And Taylor. Taylor can be your phone a friend. Same thing with Jerry, right? Okay. So why won't muscular contraction occur if the myosin ATPase activity is rendered inactive? What can't you do? Okay, so that's kind of like the end step, but what happens that causes you to not bind actin? What can you not do? Can someone say it? You can't, okay, no, not the H band, has nothing to do with the H band. What?
Can't produce ADP.
How do you, what's the technical term to produce ADP? Starts with an H. Hydrolyze, okay? So again, the answer, if I ask you that question, the answer I'm looking for is, I know skeletal muscle contraction isn't gonna occur because here's the thing. Anytime I ask you questions like that, two things will happen. It'll either not work or it'll work a little too well that you can't relax. Those are the only two scenarios. We all obviously know this one's not gonna work, right? But why not? Because if you render your myosin ATPase activity inactive, you can't hydrolyze ATP into ADP and a phosphate group. And if you have no ADP, what can you not form? You're skipping a step. What can you not form? Cross bridges, which is where you can't attach your myosin head to, of what protein? Actin. Okay, let me explain that again. If you render your myosin ATPase inactive, that means that you can't hydrolyze, you cannot split what into what? ATP into ADP and a phosphate group. And if you have no ADP, your myosin head can't attach to actin. Can't attach to actin. Meaning you can't form something called a cross bridge, which you'll hear me say later on. If you can't form a cross bridge, there's no muscle contraction. Yes? There's another question for you. I mutated tropomyosin so that its affinity for actin has increased to 1,000% more than normal. How would that affect muscular contraction? I increased the affinity of tropomyosin for actin to 1,000 times more than normal. Affinity means how tightly it binds actin. How would that affect your muscle contraction? Okay, will it work or not work?
Work.
It won't work.
Okay, I want you guys to think about it, okay? The question that I asked you is if I mutated tropomyosin to where it loves binding actin so much, it loves it 1,000 times more than normal. Here's the analogy. You guys ever watch those Lifetime movies where it's always a woman scorned or a guy scorned? If I can't have you, no one can. Right? It's all to the point of obsession. Okay. So what's gonna happen here, where if tropomyosin loves actin so much, it loves it 1,000 times more than normal, what will happen? Can you have a contraction or not?
No.
Why not? Because what?
The gate won't open.
What?
Because the gate doesn't open. Right?
Okay, in more technical terms.
The
You can't do what?
tropomyosin.
You're correct,
The
but I need,
You can't
you can't move what?
The tropomyosin, that's it, right?
Yeah, tropomyosin.
It won't be able to move because you don't have the acid leaf, right?
Close, okay? So she's halfway there, okay? Muscle contraction won't work because if the affinity of tropomyosin is increased to 1,000 times more than normal, is it gonna bind it more tightly or less tightly? More tightly, to the point where I want you guys to imagine this. If you have a gate, you welded that gate shut. If that gate is welded shut or rusted shut, can you move it? No, and if you can't move it, if this can't be moved, then what's the point of troponin binding calcium? Can it open it? Right? Even if you have a key, if it's welded shut, you can't slide it, right? And if you can't slide it, what do you not expose?
The myosin.
And if there's no myosin binding site available, can you form cross bridges between myosin and actin?
No.
No, you can't. What? Any questions on that? See how I can ask you multiple questions depending on the different parts? And then you really have to think about it. And like I said, you can tell me, oh yeah, muscle contraction doesn't work or muscle contraction will proceed, but will I give you credit for those answers? No, because did you adequately explain the process to me? No. So notice how I'm kind of giving you a way into Oleli's thinking here and what I'm looking for. So should you give me answers like that for your lecture exam? Okay, all right, last slide and then I'll give you guys a break. Cool. All right, so this one's kind of quick and easy. I want you guys to look at these two sets of sarcomeres here. Here's the micrograph, here's the actual diagram. Do you guys notice that on the right here, notice how you have the Z disc, and the Z disc is in the middle of what band? The I band, which is what protein only, actin or myosin? Actin only. okay? Here's the A band. During muscle contractions, the A band does not change in length. It doesn't shorten. Why not? Because are we changing the length of myosin? No, we're not. But what should disappear? The H bands, okay? Should your I bands also decrease in size? I want you to look. What's happening to your I bands? They're shorter because what's gonna happen here is, yeah, the A bands don't change in length, but they become closer to each other because you're decreasing the size. And I want you to look here. Where are you moving actin again? Towards what? Towards the center, towards that M line, right? And because you're sliding it towards the center, that zone of overlap is gonna increase, which means that your I band is going to shorten. So here's the micrograph, okay? The ones at the top is where your sarcomeres are completely relaxed. Notice how you have a distinct I band and you have an H band here. Notice how this is lighter than the actin myosin overlap, but nowhere near as light as the I band. Does everybody see it? When your sarcomeres are partially contracted, the actin goes towards the center. What do you notice happening to your H bands? They're becoming smaller. What is happening to your I bands? They're also becoming smaller. What is happening to your A bands? They're staying the same, but they're getting a little bit darker or lighter? Darker. Look at the picture here. Notice how they're getting a little bit darker, right? Why? Because that H band is disappearing. And if you're fully contracted, you now have a full overlap. What happened to your H band? Is it still there? No, it's completely gone now. You don't see it. What happened to your I band? Way smaller now. So now your two A bands, which are still the same length, are now closer to one another. Everybody here clear? Yes? Okay. I just need two people, and you have to be okay with me touching your hand. No one wants to volunteer. I have no cooties. Thank you. And then I think you already, I'm gonna zap you later. Andrea, come on up, ladies. What are these markers? ATP. Okay, so I need one of you guys to stand on my left and the other one on your right. Okay, so I want you to straighten your hand out so that the only thing I can grab, is it okay if I touch your hands? Okay, the only thing that I can touch is your fingertips like this. Everyone good here? Okay, so I want you guys to imagine that I am myosin, these ladies are actin. Everybody here good? Okay, I have ATP in my hands. Can I form a cross bridge with them? Can I attach my myosin heads to them? Yes or no? Yes. ATP in my hands? No. What enzymatic activity do I need to activate to let me touch them? Myosin, ATP, ACE. Okay, you guys need to remember that. I need to activate the myosin ATP ACE activity in my hands for me to do what with ATP? Starts with an H. Hydrolyze it from ATP into ADP and a phosphate group. So I want you guys to notice how I'm still holding it like this. So at this point, I'm really sorry if I get ink on you, but these are washable markers, okay? So notice how at this point, because I now have what in my hand? What's the marker part? ADP. Notice how I can now connect my hands with theirs. Everybody sees that? That is called a cross bridge, a myosin cross bridge, okay? My heads are attached to actin. Everybody good? Okay. I will now get rid of the caps. And when I get rid of the caps, I've just gotten rid of the phosphate group. Getting rid of the phosphate group releases what? Energy. Energy then that I can use to cock my head and induce something called a power stroke. Everybody see it? What's this band called where it's just myosin only? H band. What happened to my H band right now? It got smaller. Okay. What's in my hand still? ADP or ATP? ADP. For me to detach, don't move back, okay? For me to detach, what do I need to replace ADP with? ATP. Okay, so stay where you guys are at. Notice how I now have ATP in my hands. Notice how I am no longer touching them. Everybody here good? What enzymatic activity do I need to turn on again? myosin, ATPase. What do I do with these guys? Hydrolyze it into ADP. ADP and a phosphate group. So at this point, I now have ADP in my hands. What can I do now? I can form another cross bridge, but notice where I am now grabbing them. Am I grabbing them at the handsaw, or am I grabbing a little bit farther back? Farther back, closer to their axillary region, right? Okay, so at this point, I am grabbing, I will get rid of the phosphate group, and because I have ADP still, and I got rid of the phosphate group, what did I just do? Power stroke. For me to release, I need to get rid of ADP and replace it with ATP. Does everybody understand that? Notice how did my length as myosin stay the same? Yes or no? Yes, it did. So that's my A band. What disappeared? The H band. Good. Thank you, ladies. Does everybody understand that?
Yes.
Take a break. I need to zap somebody.